Hello, everyone. I'm Sylvia Lee, and I want to welcome you to the Diatom Web Academy, brought to you by the Diatom Taxonomic Certification Committee of the Society for Freshwater Science and diatoms.org. You can check out the news page on diatoms.org for the schedule of webinar speakers and follow us on YouTube, Twitter, and Facebook for updates. We are still looking for a webinar speaker in August. If you would like to be a speaker, please email us or let us know in the chat. Our next webinar will be on Tuesday, June the 8th at the same time on the genus Diploneus. So with that, I will hand it over to Sarah to introduce our today's speaker.
Thanks, Sylvia. And today we have Dr. Eveline Pinseel. Eveline is an evolutionary biologist with a PhD from Ghent University in Belgium. Her research is directed at answering questions of protist diversity and evolution using a variety of approaches from field collections, laboratory culturing, morphological observations, molecular sequencing, modeling, and she works from the Arctic to the Antarctic and all the lands in between. Eveline is currently a postdoc in Andy Alverson's lab at the University of Arkansas, and she has funding from the Simons Foundation. Her current work is to work to better understand how marine diatoms adapted to freshwater environments over evolutionary timescales by interpreting transcriptomes. Today, Eveline will present her talk using molecular phylogenetics to study diatom diversity and biogeography, Pinnularia borealis as a case study. Thank you, and here's Eveline.
Hello, everyone. Thank you for joining today's webinar. The next 45 minutes or so, I'm going to give you an overview of how we can use molecular phylogenetic data to get information on diatom diversity and biogeography. And most of the work I will be presenting was the work I did during my PhD at Ghent University and Massey Botanic Garden. And this talk is going to give you an overview starting from going into the field, finding live diatoms, culturing diatoms, sequencing them, and finally answering questions on diversity using molecular data. Okay, so I'm just going to start with a brief introduction on the genus Spinellaria, which is all about in this presentation. And this is actually one of the most species-rich diatom genera out there. So Diatom Base lists about 3,600 taxa, but this also includes a lot of varieties and formats. On Algae Base, we find 1,400 names, more or less, and about 800 of these are currently taxonomically accepted. So it's a really diverse group of diatoms, which we typically find in freshwater environments. For example, the littoral zones of freshwater ponds and lakes, rivers, wetlands, etc. And some of these species in this genus also occur in more terrestrial environments. And morphologically, they really are characterized by having these linear lanceolate valve outlines with broadly rounded apices, which can sometimes also be a little bit pointed. One of the major characteristics of the genus is the fact that the striae are chambered. So they are called the alveoli. On the outside, they are covered with a plate, which is perforated with small pores. And on the inside of the valve, this plate, this alveolus, this chamber has at least one opening that can also be entirely open, as is seen here. And this is one of the major characteristics for this genus. Based on molecular phylogenetic data, we know that the origin of this genus lays about 60 million years ago in the late Cretaceous. And the genus, in fact, is paraphyletic because there are some other diatom genera, Calonaeus and Pinnunavis, for example, which cluster in the middle of the Pinnularia clade. And this indicates there is really still a lot of higher order classification work to be done in this group. But in this presentation, I'm just going to be focusing on one member of the genus, Spinellaria borealis. And this is one of those Spinnellarias which typically lives in terrestrial environments. So it prefers soils, mosses, which can be wet, moist, or even really dry. They can occur on tree barks, they can occur on cave walls, etc. They rarely occur in littoral zones, so submerged environments, but if they do, it's more, we more often find them in these conditions in the Antarctic region, where they more often occur really in littoral zones. But everywhere else in the world, they really are truly terrestrial. We find them everywhere. This group is considered to be cosmopolitans. We find them in tundra vegetation in the high Arctic. We find them in the polar desert of Antarctica. We find them in temperate regions. We find them in tropical regions. And we even found them on the most remote island of the planet, Bouvetøya or Bouvet Island, which is located in between South Africa and South America. And this island is located thousands of kilometers from any nearby landmass. But still, Pinnularia borealis inhabits the shores, which have some moss patches. So literally, this diatom has actually conquered the world. But we can ask ourselves the question whether we could reasonably expect to find the same species in a polar desert in Antarctica and a tropical rainforest in Middle Africa. And indeed, if we look at what Diatom Base tells us, we see there's actually 67 varieties and forma that have been described. A lot of these are currently not used or are not taxonomically accepted, but it clearly indicates that there is some morphological variability there, and taxonomists over the last 2 centuries have been trying to classify this by using a lot of descriptions on variety and forma level. And similarly, some taxa have also been elevated to species level because they are morphologically distinct from what is considered Pinnularia borealis. So clearly, this is a group of diatoms which are morphologically pretty similar. We're not talking about one species. And to make things more complicated, Pinnularia borealis also has a larger sister, Pinnularia neorabenhorsti, which shows the same pattern. Again, we have a lot of varieties described in the literature, and also some taxa have been elevated to species level. And actually, I'm cheating a little bit here because the diatom on the top of my slide is actually not neorabenhorsti. This is Australo-rabenhorsti. And some of you might know this group as Pinnularia rabenhorsti because it has been used, this name has been used for it for more than a century already. But recently, Sokolova and colleagues found that Pinnularia rabenhorsti had been wrongfully transferred from the genus Navicola because another diatom was already described as Pinnularia rabenhorsti at the time, but was forgotten in the literature. And we recently found the type material of the real rabenhorsti, and as you can see, these 2 diatoms have nothing in common. So from now on, we should not be referring to this group as rabenhorsti, but as neo-rabenhorsti. Now, I just want to show you some of the morphological variability that we can see. So these are all pictures of Pinnularia borealis and Pinnularia neo-rabenhorsti valves from the maritime Antarctic region. And if I just indicate where the species boundaries are, you can clearly see that there is differences there between species regarding mostly valve outline and cell size. But some of these species are pretty similar. For example, variety borealis and variety scalaris, these 2 taxa are very morphologically similar. But what they all have in common is that they have these very broad striae that are distantly placed from one another. And this is a unique characteristic for all of these diatoms, which are morphologically relatively similar. And this was already recognized by Kleve, who described all of these together in Pinnularia section Distantes. And in the remainder of the presentation, I'm going to be referring to Distantes as the Pinnularia borealis species complex, because this is really on a phylogenetic level, one and the same thing. And I will show you that later. Then back in 2013, Caroline Souffreau, one of my colleagues in Ghent, she was the first to do a molecular phylogenetic analysis on Pinnularia borealis. So on the left, there are some pictures of cultures that she had of Pinnularia borealis. And on the right, we see the phylogeny that came out of that. And in this phylogeny, we have these distinct clusters of sequences that cluster together, that have high statistical support in front of them, and there is some kind of a long branch in front of them. And this is one indication, a first indication that there are multiple species present in the molecular phylogeny. So this is typically how different species will manifest in a phylogeny. Now, Caroline then looked at the link between the morphology of the cultures and the phylogeny, and she found that, for example, borealis variety borealis occurs 5 times in this phylogeny in distinct clades. And the same is true for variety scalaris. And to make things worse, one of these clades has both variety scalaris and variety borealis within the same sequence cluster. What this tells us is that there seems to be more variation present than morphology can tell us, and that morphological descriptions in the literature do not seem to fit actual biological taxa. So to really understand what's going on in this group, we might want to do much more with molecular data rather than morphological data. And this is exactly what I set out to do in my PhD. So what we did, we sampled globally. So we wanted to have as much diversity of Pinnellaria borealis and Neorabenhorstia as possible. And we sampled more than 1,500 samples, predominantly in the polar and temperate regions of both hemispheres. And this was mostly because of logistical reasons. And this sampling would not have been possible without the help of many people who have been sending me samples over a time period of about 4 years. Now, if you're in the field sampling for Pinnellaria borealis, I mean, this is not a tree which you can easily find. I mean, you have to sample blind and hope that based on the habitat type that you're sampling, that you also find the diatom that you're interested in. So here I'm showing you some pictures of localities where we found Pinnellaria borealis, just to give you an idea exactly how these terrestrial habitats look like. And typically, if you're sampling for live diatoms, you will want to do live sampling, of course, meaning you want to keep your cells alive until they arrive in the lab, and preferably also a little bit longer. In the lab, you will want to, in the field, you will want to keep your samples dark and cool. Because you want to keep your samples alive as long as possible. And by doing that, you will prolong their lifespan. Cool really depends on where you're sampling. If we're talking about tropical regions, cool will be about 15 degrees Celsius. If we're talking about temperate or polar regions, I would say around 4 or 5 degrees Celsius. And it's absolutely crucial that you do not expose your samples to direct sunlight when in the field, because if that happens, it could kill your diatom community really, really rapidly. And we always have to think about oxygen. You do not want your samples to become anoxic during transport. So it's always best to keep at least 1/3 of the recipient empty. And to also not add any chemicals, including culture medium. Culture medium will not help your diatoms inside. It will just increase the chance of getting anoxic conditions. And if you want to keep your samples alive, you should not add any chemicals like ethanol or anything else. However, if you want to do single cell analyses, then it could be beneficial to add some, for example, RNA later as a preservative, but otherwise I would not add anything. And then typically when your diatoms arrive in the lab, what I do, and the time between sampling and arrival in the lab, it could be a few days, it could be up to 6 months. And when they finally arrive in the lab, I would typically take different parts of an environmental sample and incubate it in a 12-well plate, as you can see here. And then I would start screening those samples repeatedly to see if I could find Pinnellaria borealis in my case. Sometimes you find them immediately. Sometimes it takes a few days, or you find empty valves first, which is a first indication that there at least has been a population present at some point. And in the most extreme case, I had to wait 1 full year before I found the first living cell in a sample. So sometimes you have to be patient. And then when you find your living cells, you will want to do a single cell isolation. And you can do that, for example, with the drop method, which was actually nicely demonstrated by Paul Hamilton last week, 2 weeks ago. So what I typically do is you have a glass pipette and you will transfer your cell from one drop to another to clean it during several transfers. And this method is most widely used by most labs. It's easy to use and easy to learn. But I actually made use of another method, which combines an insulin needle and an automatic pipette. And the advantage of this method is that you do not have to transfer your cell from one drop to another, so it's much faster. But you can only use it for benthic diatoms. So if anyone's interested in this, I can definitely give you a protocol on how to do this exactly. Now, when you have your single cell, you will typically put them in a 96 well plate, such as here, for example. And each of these wells will contain one cell, and you will start monitoring them. For example, if there is contaminants present, like another algae or another diatom, you will want to do a re-isolation. It really has to be clean. Bacteria generally are not a problem for sequencing. They might overgrow your cells. So if there's too many of them, you could do, for example, antibiotic treatments, but usually I would try to not do that. And then during subsequent weeks, you will start upscaling. So you go from a 96 well plate to a 24 and finally to a 6 well plate. And during this time period, it is really important to know the preferences, the culture preferences of your diatom. For example, light intensity is important, day-night cycle, temperature, and culture medium. So specifically for Pinnularia borealis, for example, I noticed that it prefers growing in indirect light rather than giving it a lot of exposure to direct light, as would be beneficial for most diatoms. So it really depends on the diatom you're working with, and you might want to shift a little bit with different conditions to see what works best. And if you don't have enough biomass in your 6 well plate, you can typically take a morphology sample. For example, you can take a microscopy slide, the way you would do for any environmental sample. You can also prepare steps for SEM. A second, you will want to take a DNA sample. And you can do that by storing your culture at -80°C. And after that, you can do DNA extraction, you can do PCR and sequencing. And then you can do 2 things. You can either throw away your culture, which you have been growing because now you got your DNA and your morphology, or you can think about keeping them for a while in the lab. What I typically recommend if you want to keep a culture for several years is to do that at low temperature as possible, low light, and using a small inoculum, because diatoms will decrease their cell size during their cell cycle, meaning that if your culture becomes too small, it will become inviable and you will lose it. So you will want to postpone this moment as long as possible by slowing them down. And if you're lucky, you can also use cryopreservation. So cryopreservation is a method where we keep diatoms at ultra low temperatures, so between -160 and -180°C, usually with adding a cryoprotectant substance such as DMSO. And these type of methods will mostly be useful for marine diatoms, but they also happen to be really useful for borealis. And if you're interested in this, if you have cultures that you're interested to keep long term, I would definitely recommend to contact the culture collection that was present in the lab where I did my PhD, because they have extensive experience with these type of methods for long-term preservation of diatom strains. Now I have all of these samples in the freezer. I got all of these slides of my culture somewhere in a closet. And now the exciting part starts. I want to do molecular species delimitation. And the first thing you have to do if you want to do this is you have to choose which markers to sequence. And here on the left, I'm showing you a list of the most general markers which are sequenced for diatoms at the moment. So these includes nuclear genes from the ribosomal DNA, chloroplast genes, and mitochondrial genes. And if you choose a marker, it really depends on what you want to do. For example, if you want to identify species, 1 base pair difference between 2 markers is going to be sufficient. If that base pair difference is consistent between 2 species, that's enough. However, that is never going to be enough if you want to delimit species. If you want to have really confidence in your species boundaries, in your species limitation, you will want to have variable markers which differ a lot in their sequences between species. And if you're interested in phylogenetic relationships, you will typically want to include multiple markers, because different markers will have different resolutions in different parts of your phylogeny. So by combining information of multiple markers, you will get a much better sense on phylogenetic relationships. Now in diatoms, 2 genes have been most routinely sequenced up to these days. And this is the chloroplast gene RBCL, which codes for the protein rubisco, and then the 18S, a small subunit from the nuclear genome. And RBCL, I think in general, for most diatom genera, it's a really good first choice. It's relatively variable. It's probably very good for identification of species, and most likely in most diatom genera also for delimitation of species. There's a lot of reference sequences available on GenBank, and it's easy to sequence. So it has a lot of advantages. 18S has many of the advantages of RBCL. There is a real ton of reference sequences on GenBank. It's probably the most sequenced gene for protists. The problem is that we know that it probably, well, it for sure cannot discriminate between closely related diatom species in many complexes. So I've seen this happening in the Eunosia bulinaris complex, in the Agnanthidium minutissimum complex, and it's also true in Pinnularia borealis, that different species necessarily do not differ for their 18S sequence. So even though this gene is widely used, I would really recommend to be careful with hinging too much interpretation on this gene, because it's not that variable. And if you want to know more about like marker resolution and which markers to choose and disadvantages, advantages between markers, I would still recommend the paper in Protest by Andy Alverson from 2008. It's more than a decade old, but I guess it's still a very good read if you are interested in molecular systematics and diatoms. And you really have to keep into mind in all cases that there is going to be differences between genera. It's not because a marker works in one genus that it is going to work in every diatom genus. So you always have to be critical when you start working on a new group, whether your marker is really showing what you want it to show. And I want to demonstrate this with the Pinnularia giba group, which is another clade in the genus Spinellaria. And this is work done by my colleague, Jan Kolar from Czech Republic. And Jan sequenced a whole bunch of species of the Pinnularia giba group. You can see some of them here on this picture on the right of the slide. And he sequenced multiple genes of these. And then for each of these strains, he actually looked at the differences in variability between two markers. So I will start with the first one here. And here he's comparing 18S. So the small subunit ribosomal DNA. And it's actually only comparing the V4 region, which is the most variable part of the gene, with mitochondrial COX-1. And if a sampling point would be on this line, it would mean that for one given strain, the sequence is equally variable in 18S as it is in COX-1. But you can see that most of these sample points actually plot above this line. And what this tells us is that COX-1 is much, much more variable than 18S within these species of Pinnularia giba group. He then compared RBCL, the chloroplast gene, with 18S. And here we see a pattern where most of the sampling points plot above this line, indicating that 18S is more variable than RBCL. And with this, I want to show you there's differences between genera. In most diatom genera, you're going to see the opposite pattern. So RBCL is supposed to be much more variable than 18S, not in Pinnularia. In Pinnularia, 18S is much more variable, and RBCL is not a good choice for species limitation. Now, when combining all of these results, we actually found that if we want to do species limitation in Pinnularia, two markers have a lot of promise, one being mitochondrial COX-1 and the other being the 28S, so the large subunit of the ribosomal DNA. And in fact, if we compare these two, it looks like 28S is actually even more variable than COX-1. So we have two really variable markers here. And 28S, it's great. There is quite some reference sequences. It has been relatively routinely sequenced. There's really good primers. It's easy protocol, so easy gene to work with. COX-1, not so much. The problem with COX-1 is that there are actually very few standard primers available, and those that are available are not universal. So they cannot amplify any diatom you want. So they only work on a few diatom groups. And unfortunately, they also do not work on borealis, although they work on most other Pinnularias. But there is actually another primer pair in the literature, which has been forgotten, and which has the same problem as the universal primer set by Saunders and Evans, but it has a slightly different resolution in different parts of the diatom tree. So sometimes it's able to retrieve sequences where the Saunders and Evans pair is not able to do that. And using this primer pair, I was able to get a few sequences of Pinnularia borealis COX-1. Efficiency was still really low, but I want to show you because as soon as you get maybe 4 or 5 sequences out from different species, that's enough to develop your own primers. And that's exactly what I did. So I came up with 6 different primer sets for COX-1, 3 of which I started routinely using in my own research because the other 3 weren't really great. And the fact that I needed to use 3 primer sets immediately underscores the variability of this gene because it indicates that even in these primer regions, a lot of mutations are happening, meaning that it's extremely variable and therefore a really good choice for species limitation. So even though if you're working on a group and your COX-1 primers do not work, and that is very likely to happen, I would say do not panic. Start to try with what else is present in the literature, and you might be able to actually design primers for your group, and you will have much, much better sequence data than when using, for example, 18S as your general marker. And now as a result, I actually had a bunch of sequences, more than 800 strains sequenced of Pinnularia borealis for 28S and COX-1. And what you then typically do is you make an alignment of your data, as shown here on the top left, and then you make a molecular phylogeny out of that. And what we typically do in the diatom literature, we do a visual inspection of our phylogeny to assess where are the species boundaries. So where do we see different species present? As I said before, these typically are these clusters which have large statistical support with a relatively large branch in front of them. That's the first indication of the presence of a species. But if you have a really big data set with lots of closely related species, this is actually going to be a very subjective method to assess species boundaries. At this point, it might be better to actually do this more in an automated way. And there are solutions to this. So there is a whole bunch of literature, mostly developed on macrobiota, where people have designed methods to automatically delimit species in sequence data. So they can use these methods, use your alignment, they use your phylogenies, so they can use different sources of your data, in all cases molecular data, and they will try to assess species boundaries automatically. And these methods, they have been used really widely on macrobiota. They are often used as a first indication for different species and even for species descriptions. They are, for example, becoming much more of the golden standard even in seaweed taxonomy. So in seaweeds, these methods are applied all the time. But for some reason, it has never gotten any traction in diatom as systematics. So what Jan and I did, Jan Collard and me, we decided, well, let's just select several of these methods which are really useful for the type of data we have, and let's apply this on both Pinnularia borealis and Pinnularia gibba. And I'm just going to show you the results of Pinnularia borealis, but if you're interested in gibba, I would definitely recommend to read Jan's paper in Journal of Ecology. So here I'm showing you the results of these automated species limitation methods using the 28S gene. So this is a phylogeny that I'm showing you here, which was based on the 28S gene, but I'm just plotting it in a circle because otherwise it wouldn't fit on the slide. And the different colored bands, they actually indicate the results of our five different species limitation methods, which are here indicated in the legend. And those white kind of boundaries, they indicate where the species boundaries are. So if you look in this part of the phylogeny and over here, it actually looks like these methods really agree. So they delimit exactly the same species within Pinnularia borealis. But then there are some parts, for example, here and here, where some of these methods disagree, and they're starting to delimit a different number of species. And I'm showing you this because none of these methods are a golden bullet. They just give you a first indication of where species boundaries are, but it's always a good idea to combine multiple methods and to look for consensus among them. So if you look at the number of species delimited by these methods in the 28S gene, we have between 88 and 98 different species. So these numbers are close to one another, but there are actually some differences there. Now, what I then typically do is I use a majority rule. So I say, well, in order for one of these species to be accepted as a species, I actually have to find it in at least 4 out of 5 methods in order to accept it. So doing that for 28S, we found that there's actually 84 that could be classified as a species. Now, we did the same thing for our mitochondrial COX-1 data, which are, as I said before, extremely variable. And if you look at the results, we see that, well, we have a much larger number of potential species limited here. And also these numbers are actually much closer to one another than for the 28S gene between those methods. And if you look at the consensus species limitation, we could accept 126 as the number of species in our data set, or potential species. Now we have a problem here. We have 2 molecular markers, and both of them indicate another consensus species limitation. Only one of them can be closer to the truth than the other. So what's going on here? And what I then like to do is actually I like to try to see if I can find additional evidence to see which of these markers might be much more inclined to tell us what the more or less the real species number is. And I said before that in the phylogeny of Caroline Souffreau, all of these clades were morphologically very similar. But I also said that some of the taxa in Pinnularia borealis and Neorabenhorsti are morphologically distinct. So by looking for those morphologically distinct taxa in our big data set, we can actually assess which of these methods is more likely to be correct. So yes, morphology can inform molecular species limitation, and I'm just going to show you how. So here I'm showing you a lot of closely related Pinnularia borealis and Pinnularia neorabenhorsti sequences. And according to 28S, all of these, which is this dark gray bar, which indicates delimitation by 28S, according to 28S, all of this is still one and the same species. But COX-1 says no. COX-1, which are these little gray boxes, it says no, there is actually 7 different species. Each of these clades is a different species. So I looked at morphology, and if you look at the top 4 species, they are morphologically very similar. I mean, you could argue that the top one has some difference in length-width ratio compared to the other 3, but in general, they are morphologically very similar. But if you look at the bottom 3 clades, these taxa are much, much larger. And not a single morphotaxonomist would put these large-celled morphotypes together in the same species as these small-celled morphotypes. To make things more complicated, one of the clades actually has spines, and these spines are visible in the light microscope, and they are pretty beautiful if you look at the SEM. And we find these spines both in cultures and in natural populations, in natural samples. So they definitely are there, and they are consistently present in this species. So what we actually see here are distinct morphological differences. COX-1 was able to recognize these clades as different species. 28S was not. So we actually decided that COX-1 is much more likely to show us the real species number and not 28S. This does not mean that these species do not differ in their 28S sequence. They actually do, but they just don't differ enough for these automated molecular species limitation methods to recognize them as different species. And this is why you really need those really highly variable markers before you can actually do these type of methods. So this is not going to work on a marker like 18S or RBCL in Pinnularia, at least. And to demonstrate that, many of the clades in here actually have identical sequences for 18S. For example, the large cell species with the spines has exactly the same RBCL sequence also as this Pinnularia borealis morphotype. And nobody would ever put that in the same species. So just to always be critical about the resolution of the marker and the group you're working with. So we actually came up with, okay, we have 126 putative species level images. There might still be, the reality might be a little bit different, but this is more or less the magnitude of the species we're talking about. And the minimum difference between all of these species in COX-1 is at least 11 base pairs, which is quite a lot, actually. And in fact, if we want to understand how many species we could expect on a global scale, because of course we haven't sampled large parts of our planet, we extrapolated our data set and we found that, well, using these type of methods, we could expect to find more than 400 species on a global scale, potentially even more, potentially a little bit less. This is a relatively rough method for extrapolation, but it tells us at least we're not even halfway at finding the diversity out there. And interestingly, if we look at this phylogeny, where I've indicated for each of the species one light microscopy picture, we actually see that these large-celled morphotypes, which represent Neorabenhorstii morphotypes, that these have evolved multiple times. And that's what I meant with Pinnularia borealis and Neorabenhorstii. They're one and the same thing. They actually belong to one and the same monophyletic clade. So they're not two distinct clades in the genus Spinellaria. But many of these taxa are morphologically very, very similar. And we can start asking the question like, okay, exactly how similar are these? Because our brains are not always that good at telling morphological differences, especially if it's differences regarding shape or valve outline. So we did a morphometric analysis because often computer programs are much better at trying to find subtle but consistent morphological differences. And I did that in collaboration with Jana Kulichava from the Charles University in Prague. And we used Sherpa by Michael Kloster as our program for the morphometric analysis, because Sherpa is really good as well. It's very easy to use, and it's really good at getting information on shape from pictures. And we also looked at ultrastructure, so we did a lot of SEM, and for example, we quantified the density of the areolae in the alveoli as a parameter. We even looked at chloroplast arrangement morphology and nucleus morphology, because, for example, for the Navicula cryptacephala complex, we know that different species tend to have different nucleus morphologies. And doing all of that, we find that, yes, there are, between the 8 species we selected, we actually find subtle morphological differences. And these differences relate to the outline of the valve, cell size, the width of the axial area, and the density of the areolae. And if we look here on the left, we have our multivariate plot, and then we actually see that yes, some of these species are clearly distinct, like the yellow, the purple, and the blue. We can clearly differentiate them from one another. But unfortunately, there is a lot of variation within species, and a lot of these things are also overlapping. So on average, we can define the morphology of each of these species, and on average, these morphologies are significantly different from one another. But the fact that there is so much variation and so much overlap between species makes it very difficult to actually use morphology in the end to assess species boundaries. And we also found this when we were actually doing a classification model. So what we did was we had a model which was trained on Pinnularia borealis data from our previous 8 strains. And here on the y-axis, you can see the accuracy of this model in percentage. And on the other y-axis on the right, we can see the number of species that were included in the analysis. And on the x-axis, we see the number of strains. So if you give this model just like 2 species and only a couple of strains, its accuracy of classifications is really high. But as soon as you start adding more species and more strains, our accuracy is dropping significantly. What this tells us is that the more information this model is giving, the poorer it becomes at discriminating between species, simply because there's so much morphological variability present. And this is only with 8 species. So imagine what happens when you have like 50 or 100 out there. It's definitely not going to work anymore. This doesn't mean that morphology cannot be useful. As I said before, from time to time, species have distinct morphology. They are easy to recognize and can even be used to inform species limitation. So it's never just an easy answer of yes or no. It's always like a gray zone of maybe or maybe not. Now you have all of these species, and now you can actually do the most exciting part. You can start looking at evolutionary history and biogeography. And a starting point for this with molecular data is, first thing you need to do is obtain a species tree. So a species tree is a phylogeny that will give you the relationships between all the species that we delimited. So remember, we have 126 species, and for each of these, I selected 1 strain. And for all of these strains, so 126 strains, I sequenced 6 genes. So 2 nuclear genes, in this case 18S and 28S, 3 plastic genes, and 1 mitochondrial gene. And these different markers have different resolutions in different parts of the trees. We have very variable markers such as 28S, COX-1, and more conserved markers such as PSVA and PSVC, which are very conserved, and 18S and RBCL somewhere lay in the middle. So this gives us different resolutions in different parts of the trees, which should help us to get a good species tree. And this is the result. So this is the species tree of Pinnularia borealis. And we have a large outgroup included in the analysis, which includes other species from the genus Spinellaria and also Celophora and Maiamea, which are sister genera. And this, with the black bar, this indicates the borealis complex. And as you can see, these are actually 5 different clades, and there are no morphological differences between those clades. So we find the same morphologies in these different clades, both large and small cell species. And this phylogeny is actually a time-calibrated phylogeny. So the x-axis indicates time, so 1 million years before present. And how we get those typically is by using fossil data from the literature to calibrate our phylogeny. So fossil data, if they have a good age associated with them, and if we can assign them to well-known nodes in the phylogeny, we can use those fossils to get information on the age of our phylogeny. And I typically do that in BEAST, but there's actually a whole myriad of programs that can do this using different types of algorithms. And if you then have your time calibration phylogeny, and this is basically the same one as the previous slide, I'm just showing you only the borealis complex to make things easier with the 5 different clades. We see that its diversification started between 25 and 36 million years ago. So this was a really interesting time period because this is the boundary between the Eocene and the Oligocene. And this period was characterized by the last shift from our planet from a greenhouse to an icehouse state. So at this point, temperatures globally dropped substantially, never really recovered. And we saw associated with this also an opening up of landscapes, especially during the Miocene. We had tundra vegetation developing, we had grasslands developing on a global scale. And these are typically the habitats where borealis is easily found, and disturbed habitats, for example, disturbed by large grazers or disturbed by humans. So it looks like that this shift to a more cooler planet might have actually benefited this diatom in some way. Now, if we have a time-calibrated phylogeny, you can also start understanding ecology. For example, you can get information about the ecology of ancestral species. So what we did, we actually looked at the current habitat type of each of our borealis species, either terrestrial or aquatic, because some of these species are confined to aquatic environments. And then doing that, we did an ancestral state reconstruction, which allowed us to see where the shift to this predominantly terrestrial habitat happened. And it seems that this happened at the start of the diversification. So also at the Eocene-Oligocene boundary is when the ancestor of this clade shifted from a more freshwater lifestyle typical for Pinnularia to a clearly terrestrial lifestyle. And this shift is associated with extreme thermal tolerance. So we know from experiments that borealis survives minus 180 degrees Celsius. So you can literally drop it in a cryo vessel without any cryoprotectant and the culture will come out alive. And this happens for both polar and temperate strains from our different clades. And they're also very tolerant to desiccation. Again, members of different clades are tolerant to desiccation. And this suggests that these characteristics such as extreme temperature tolerance and desiccation tolerance are ancestral and likely evolved also when the group shifted to this terrestrial lifestyle. You can also use a molecular phylogeny such as here to understand biogeography. So here I'm showing you a map of the world where we indicated with different colors different biogeographical zones, which we delimited for Pinnularia borealis. And then here on the right, all of these boxes correspond with this same biogeographical zone. So each of these branches is one species, and each of these boxes indicates where each individual species was found. So you can see that most of these taxa seem to have only one box colored, which means they only were found in one of these regions, meaning they have restricted geographic distributions. Of course, we did not sample the whole planet, so some of these might actually have much wider distributions. There's definitely sampling bias here, but it's quite striking that the majority that we found seem to have very restricted distributions. Some are actually cosmopolitan. So some species we found in both the northern and the southern hemisphere, they seem to be widely distributed. One of these is actually also very dominant. It's actually most likely, if you find Pinnularia borealis, it's going to be this species, which seems to be pretty common in many samples across the world. So we have a combination of taxa with very restricted distributions and some species which are widely distributed. And also we see repeated colonization. So we have our 5 clades, but it seems like, okay, each of these clades has Arctic species or Antarctic species or European species or whatever. And it actually indicates that this group has managed to colonize these regions multiple times. And it means that borealis is if it has enough time, it's actually pretty mobile, but it's not mobile enough. So gene flow is not sufficient enough to prevent diversification. Otherwise, you would not have this many different species. You can also analyze biogeography in a much more controlled way, for example, by using the R program biogeobase. And biogeobase uses the extent distributions of species to assess the distributions of the ancestors. And by doing that, it can give us information on what type of speciation events have been important in the group. And this is a typical result of biogeobase. You don't have to actually get all the details here. Just know that these bar plots indicate the different type of speciation methods or methods at which biogeography changes of species. And the more we are to the right, the more important they are. So these 3 are not really important, and these 2 are very important. Now we found first that founder event speciation is important. And what this means, and it's actually depicted here, it means that a species, for example, disperses from one region to another, for example, from a continent to a remote island. It becomes isolated at this island, and if this isolation lasts long enough, it will develop in a new species. So this is a form of allopatric speciation, and it seems to happen quite dominantly in Pinnularia borealis. We also found that within region speciation, which is depicted over here, so speciation when the ranges of a species overlap. So sympatric or parapatric speciation can be important as well. But this has to be interpreted with care because our biogeographical zones were so large that this could still include a lot of founder event speciation, but we will still not be able to recognize this with this analysis. And last, we found that there is a significant distance effect, meaning there is an inverse relationship between the distance between two regions and the probability of dispersal. So the further two regions are apart, the smaller the chance that borealis will disperse from one region to another. So dispersal is limited, and this is one mechanism at which we get so many different species. So it looks like borealis has what we call an ecological key. It has transitioned from a freshwater to a terrestrial environment. And associated with that, it developed a whole bunch of ecophysiological adaptations which allow it to survive in terrestrial environments. So extreme thermal tolerance, extreme freezing tolerance. And most likely those same adaptations also help it to survive long distance transport. If you're better at surviving freezing, you're most likely better at surviving being transported by wind over vast geographic distances. But at the same time, dispersal was not frequent enough to prevent diversification given enough time. And we think this is probably because borealis is locally rare. So although they are globally distributed, they rarely have large population sizes. They have population sizes which are much, much smaller than, for example, marine phytoplankton. And being rare means that although you're good at surviving dispersal, it means you will simply not have that many dispersal events to start with. And combined with having a very large planet with lots of remote islands, remote areas, which all have niches that could be beneficial for borealis, means that we have a good recipe for what we call a geographic radiation. So geographic radiations have rarely been studied. We know they happen based on research on fish and salamanders. And in these type of biota, they typically happen in geographically complex areas. So I'm talking about mountain regions, I'm talking about archipelagos, etc., where they typically have a lot of geographic isolation going on a small spatial scale. We think that for borealis, the same happens, but on a planetary scale. So we have so many remote areas that are very distant from one another. For example, all of these different islands, which have terrestrial habitats, which are beneficial for borealis. So having all of this terrestrial complexity on a global scale is driving diversification. This doesn't mean that, for example, borealis cannot be classified as an adaptive radiation. I definitely think that adaptation plays a major role as well. And often many radiations in macrobiota are a combination of different factors, which can be both local adaptation and geography. But I just want to highlight here that in terrestrial diatoms, we should definitely not underscore the importance of geographic isolation in speciation processes. So to conclude, we actually found that, yes, using molecular data, our diatom complex Pinnularia borealis is extremely diverse. Its diversity has been severely underestimated using morphological data. And it could very well be that we find very similar patterns for many other diatom species complexes as well. We know many other candidates out there for which research has been done, which suggests that probably we're talking about very similar diversity levels as Spinellaria borealis, if we would do the same scale of sampling. And second, it looks like that allopatric speciation can be a very powerful mechanism to generate species in terrestrial diatoms, and very likely also in fresh water diatoms. So geography definitely matters also in tiny diatoms. And with that, I want to thank you for your attention. I also want to thank all the people over all these years that have been helping me to get this research, to make this research possible, really. And if you have any questions, I would be really happy to discuss that with you.
Thank you, Eveline, for a really excellent presentation. Let me see. Oh, I'll open up my chat window. If anybody has questions, please go ahead and put them in the chat box. I wanted to just say I'm really fascinated by your time calibration work, and being able to do that. I don't know that anyone's done that so far for other groups. Has there been that kind of work on any other taxa?
Definitely. So I've been, most of my work was actually based on the time calibration by Caroline Souffreau, a paper on Pinnularia back in 2011, which was probably at that point the first time calibration of a diatom genus. So I've mostly used that calibration and updated it with new fossil data we found. And then there is some big time calibrations of the full diatom tree. The most recent one by Teofil Nakov in 2018, where they calibrated the entire diatom tree using lots of fossil time calibration points. So there's definitely some work done already. Yeah.
Let's see. No one is asking questions, so I'm going to keep going and ask. I guess the other thing I was really fascinated by was, that you talked about isolation as being so key, but at the same time, dispersal is part of this. And so it's kind of this tension between dispersal and isolation. And do you just explain that by time frame?
Mm-hmm. Yeah, so there is a big hypothesis in the literature on both fish and birds that if you have this combination of being relatively good at dispersal, but not that good that you can constantly have gene flow between distant populations on a global scale, that this is going to increase your chances for speciation. So if you're not good at dispersal, it means that you will also not be able to reach a lot of places on Earth, meaning that your chances of becoming isolated and getting a new species are small. If you get isolated, it will definitely become a new species, but your chance to become isolated initially is small. If you're really good at dispersal, you might be able to reach any point on Earth, but the fact that you can do this repeatedly and frequently will also prevent diversification. But if you're like at a sweet spot in the middle, where you're pretty good at dispersal, but not that good, so you can basically reach every place on the planet, but not frequently enough, then that's when you get a lot of species. And I think that's what's happening in this group.
Okay, Matt Ashworth, why don't you go ahead and ask your question, unmute yourself and turn your camera on. Let's see, and maybe I have to unmute you. Ask to unmute. Can you
hear me?
Yeah, got it.
All right. Hi, Eveline. That's a really interesting talk. And I actually see Mark is sort of getting at what I wanted to ask too in the chat here about scalability and how far across the diatom tree do we expect this kind of molecular diversity, right? You mentioned the Nakov talk about diversification rates being very different, once you get outside of the Raphid pennates. And there are some biological reasons to expect that, right? With reproductive biology and mating types and possible differences in dispersal rates and things like that. So, it's great to see this level of care and this level of research done on any diatom, but how do we expect these sort of patterns that we're seeing in Pinnularia borealis outside of even Pinnularia? Does that make sense?
I think so. I think there must be multiple complexes that have similar patterns. Whether the degree of diversification is going to be the same, I don't know. A lot of complexes, we still don't know really how old they are. I'm now thinking, for example, about Hanshiamphioxis, Nichia pallea. These are really good candidates for these type of patterns as well. But it all depends on how old and how long they have been around. If they only been around for, say, 15 million years, they have much less time to accumulate diversity. And I think it really relates to what Sarah just asked, like how important is dispersal and this balance between dispersal and becoming isolated? If dispersal capacity is going to differ between clades, you could expect some clades to be more diverse and others to be less diverse. There might be other parameters playing, such as local adaptation, etc. I think it's really still a big open question, a very exciting question. I think because actually I didn't show it in this talk, but I did look at diversification rates in borealis, and they seem to be higher than in many other parts of the diatom tree. But we still don't know about any other complex, whether this is the case. So there is a lot of research to be done. Yeah, but I would expect to find similar patterns at least a few more times in other complexes, for sure.
If you don't mind me asking a follow-up here, do you have any thoughts about? I mean, you mentioned Hansia, right? Do you have any thoughts about what these other complexes might be? Because I mean, sure, there's going to be a lot of diversification. The diversification is going to be higher in Pinnularia borealis because have we teased apart any other part of the diatom tree as much as this great work that you've done with Pinnularia borealis?
No,
Right.
and I guess Skeletonema has been pretty well studied as well. But in most cases, I think one of the bigger issues is that we are not always using the right markers. So if you really want to do this kind of research, we need those highly variable markers, such as COX-1 or other ones that could be available, ITS, for example. But because of like a lot of reasons, most studies focus on somewhat slower evolving markers, which will impede our ability to really discriminate really closely related species reliably. So we would really, need more work, I think, using variable markers on a global scale. That combination, what I tried here, would be able to answer that question. Yeah.
Okay.
Yeah. Maybe I'll add
sorry.
Mark Edlin's question, where he's kind of following up on this. He says 20 years ago, David Mann estimated we were severely underestimating diversity based on mating types. And your work seems to suggest that it's even more severely underestimated. What would you guess at what we might expect for total numbers of diatom species?
Yeah,
and is this pattern of undiscovered diversity similar in other groups?
I don't think I dare to put a number on it, but I would probably go more towards the higher end of David Mann's estimation rather than the lower end. And I am doubting whether the update of because the original number was 200,000 in the 1991 paper, but then there was an update, I think 2013, where they said it's more likely going to be 30,000 to 100,000. I think maybe it's going to be more than 100,000. I don't know. It could be. But yeah, it all depends on whether this pattern is going to repeat itself in many other complexes or not.
Leila has a couple of questions. What is the first geologic time point of this group?
Yeah, so the oldest fossil. Yeah, the oldest fossil is 15 million years old. It's from the Freeze Hills fossil locality in McMurdo Dry Valley sector in continental Antarctica. And at this point, we already have two species. So we have a really large celled species present and a more normal Pinnularia borealis morphotype. So this indicates that at this point of this fossil, we already had diversification. We already had different species there. But that's really the oldest time point we have. The problem with this diatom is it's terrestrial, so we have to be really lucky to find it in freshwater deposits. Really, it's always a lucky find. I think I found four valves in samples with thousands and thousands of other diatoms there. So it was a lucky find, but yeah, 15 million years.
Karthik, would you like to ask your question? I think you should be able to unmute yourself. And
hi.
Camera
wonderful talk.
Thank you.
Just so what's your experience on this cryptic and pseudo cryptic morphological diversity in Pinnularia?
You mean in Pinnularia in general, or
in borealis complex?
So my experience with like cryptic or pseudo cryptic, whether it's one of the two or like.
So do you observe like multiple taxa strains with same morphology or genetically different or genetically other way around?
Yeah, so there is a lot of species that are morphologically very similar that are genetically very, very different. Some of these morphologies have been separated for some of these species have been separated for 20 million years and still look more or less the same. And then there's things that have been separated for 1 million years and they have differences in cell size. One has spines, the other has not. So there is, sometimes morphology evolves really rapidly. And in other cases, it seems like it's really static or convergent over large timescales, unfortunately.
Okay, well, we are at the top of the hour. So I want to thank you for an excellent presentation, Eveline, and thank you to everyone for joining us today as our Diatom Web Academy continues on. And in 2 weeks, Elena Jovanovska will talk on the genus Diplones. So hope to see you all again at our next webinar. Take care until then.
Thank you. Bye.
Thanks, Eveline. Bye-bye.